Welcome to the differential gene expression module, and in this talk we're going to learn about the regulation of gene expression by controlling RNA processing. We will first learn about the splicing of RNA and the role of splicing enhancers and silencers, and then continue to learn about the role of 5' and 3' RNA modifications on gene expression.
Let us look at the different mechanisms of differential gene expression. Gene expression can be controlled at different levels, and the first level is at the transcriptional level, where we can regulate RNA synthesis and thereby control protein production in a cell. Another way to control gene expression is by controlling RNA processing. We all know that the nuclear RNA made in eukaryotic cells has to be processed to form messenger RNA, and thus by regulating RNA processing, we can regulate the protein production in a cell. Another mechanism of regulating gene expression is to regulate the translocation of the messenger RNA from the nucleus to the cytoplasm. Translation is another step of regulating gene expression because it is the step where we can regulate protein synthesis from the messenger RNA that has been made. Finally, just by making a protein does not ensure that it will actually be functional. In some proteins, post-translational modifications are required, and thus we can regulate protein function by the addition or removal of certain moieties like phosphate groups or other types of functional groups.
When DNA is synthesized into RNA, then the RNA includes not only the exons that have the information to code a protein, but they also code the introns, and these introns are shown in gray in this figure, while the exons are shown in colors. This RNA that has the introns as well as exons is called as the nuclear RNA or nRNA. In order for this nRNA to be converted to messenger RNA, splicing of this RNA precursor has to happen. Now, different combinations of potential exons can be chosen during the splicing. For example, we can have a messenger RNA that includes the exons 1, 3, and 5 that are joined together, or we can have another type of messenger RNA where we have the exons 1, 2, 4, and 5 that are joined together or spliced together. Both these messenger RNAs, even though they arose from the same nuclear RNA, they will be coding for proteins that are different from one another.
The alternative splicing of the nuclear RNA is a process of producing a wide variety of proteins from the same gene. The process of splicing requires identification of which sequences are exons and which sequences are introns. In most cases, there are consensus sequences, or sequences that are conserved at the 5' and 3' end of introns known as splice sites. So when we look at the figure below, we can see the dark green regions, which are the exons, and there are 2 of them, and the light green region, which is the intron. Right at the boundary of the 5' end, we can see a 5' splice site at the intron. This is a sequence that is normally going to provide the information that there is a boundary between an exon and an intron. Similarly, there are 3' splice sites at the 3' end of the intron.
The splicing of nuclear RNA is mediated by a complex called as the spliceosome, which is made up of small nuclear RNA or snRNA, as well as proteins, which are sometimes referred to as splicing factors. Now, cells can make different types of specific splicing factors that differ in their ability to recognize a sequence that's present in the introns. Now, here is an example where we have a nuclear RNA that has 3 exons called 1, 2, and 3. and we have introns, which are shown in gray between the exons. A cell can express certain splicing factors that mediate the joining of exons 1 and 3, and thus is what we are seeing in this picture. However, it is also possible that a cell is able to make other types of splicing factors, and thus the spliceosome is able to mediate splicing in such a way where we generate a messenger RNA which has the exons 1, 2, and 3 joined together. Thus, the same nuclear RNA is able to give rise to two types of messenger RNA. And this is an example of the cassette exon.
Another example of alternative splicing is where we have mutually exclusive exons, and this is seen in the FGFR2 gene. So when we look at this specific example, we can see the nuclear RNA having the exons 7, 8, 9, and 10. What can happen is that we can have the exons 7, 8, and 10 be joined together to form a certain messenger RNA that is observed in the limb ectoderm. However, alternative splicing can also create a messenger RNA that has 7, 9, and 10 exons joined together. In this case, we are seeing a phenomenon of mutually exclusive exons where a certain messenger RNA can have only certain types of exons. In this example, the messenger RNA can either have exon 8 or exon 9, but it cannot have both. Another type of alternative splicing is an alternative 5' splice site. In this case, we're going to look at an example where the nuclear RNA has 3 exons, exons 1, 2, and 3, but in the case of exon 2, there is an alternative splice site, and it is at the 5' end. And thus, we can form a messenger RNA where we have the entire exon 1, the entire exon 2, and the entire exon 3. However, in certain scenarios, the alternative 5' splice site present on exon 2 can be used, and it can provide a messenger RNA that has the entire exon 1, a small portion of exon 2, and the entire exon 3. This is normally observed in the BCLX gene. The third type of alternative splicing is where there are alternative 3' splice sites. In this case, we are going to look at a nuclear RNA that has the exons number 8, 9, and 10. And alternative splicing can occur where we have the entire exon 8, entire exon 9, and the complete exon 10 joined together to form the messenger RNA. However, we could have a 3' splice site being used for exon 9, which results in the complete exon 8 and exon 10 being present in the messenger RNA, but only the 3' part of exon 9 is present. Thus, there are different ways by which alternative splicing can occur. We are now going to look at different types of DNA sequences called splicing enhancers and silencers that are cis-acting sequences on the nuclear RNA that are close to the potential 5' or 3' splice sites. Cis-acting, just to remind all of you, is when the DNA sequences are on the same chromosome or on the same piece of DNA, which in this case, it can also be in the same sequence of nuclear RNA or RNA, which is what we're seeing in this case. In the case of splicing enhancers, the enhancers will promote the assembly of spliceosomes at the RNA cleavage sites. Silencers, as the name suggests, will exclude exons from an mRNA sequence. Trans-acting proteins are able to bind to splicing enhancers and recruit the spliceosomes to the area to promote splicing. Trans-acting basically means that the information for these proteins do not have to necessarily be on the same chromosome in which we can find these splicing enhancers and silencers. It should be noted that the way some trans-acting proteins can promote splicing, we can also have some trans-acting proteins that can bind to the splicing silencers and repress spliceosome formation. Now let's look at an example where splicing can actually play a role in development. Now we're going to look at a particular gene which plays a role in muscle development. So it has 3 exons, which are exon 1, 2, and 3, and it has a termination codon within the intron that is between exon 1 and 2. There's also a sequence in that DNA in the intron, which is GTAAGT, and we're going to see what happens when there is a change to that sequence. Normally, what happens is the DNA will make the nuclear RNA through transcription, and then alternate splicing will happen where the introns are completely removed and exons 1, 2, and 3 are joined together. So the termination codon that was present within the intron never gets present in the actual messenger RNA, and a protein is made, and this protein actually provides a signal to muscle cells to stop dividing, and you get a certain type of muscle that is formed, as we can see in the animal. Now let's see what happens when there is a mutation. So in this case, when you look at the DNA, that sequence that's present in the intron, the GTAAGT, it gets mutated to a GTAAAT. And the presence of that extra A actually results in a splice site being generated. Now, what can happen is alternative splicing can occur using that splice site that got generated because of the mutation. And now what happens is that the messenger RNA that is produced actually has part of the intron between exon 1 and exon 2. And this is the part of the intron that actually has the termination codon. And since it has the termination codon right in the region between exon 1 and exon 2, and it is present in the messenger RNA, the protein that is made is basically a truncated protein that does not have a function. Thus, the muscle cells do not get the signal that they need to stop dividing, and they keep dividing, and what we end up with is muscle hypertrophy, or basically organisms that have a lot of muscle than what is normally observed. Thus, we can see that how changes in splicing can cause changes in developmental phenotypes.
In addition to splicing, messenger RNA can also go through other modifications that can affect its stability as well as the ability to be translated. These other modifications include the addition of a 5' methyl cap as well as a 3' poly A tail. There have been studies that have shown that mRNA that don't have that 5' methyl cap are unable to be translated into proteins. There are also studies that have shown that the length of the poly A tail that is added to the 3' end of messenger RNAs, as well as sequences in the 3' untranslated region, are able to control the stability of messenger RNA. Now, some messenger RNAs are selectively stabilized at specific times in specific cells during developmental processes. An example of a messenger RNA that is selectively stabilized at specific times is casein. Casein is a major protein that is present in milk, and it is not expressed when a woman is not undergoing the process of lactation. Now, lactation occurs when a woman has a child or has given birth to a child, and in this instance, casein has to be expressed. So its mRNA is stabilized during lactation by the actions of a hormone called as prolactin. Thus, in this example, we can see the selective stabilization of casein at a specific time, which is after childbirth. With this, we come to the end of our talk where we learned about splicing of RNA and the role of splicing enhancers and silencers on formation of messenger RNA. We also learned about the role of the 5' and 3' RNA modifications that influence the stability and function of messenger RNA.